Oh, Good evening, everyone. President Dr. Gautam Saha, General Secretary Dr. TSS Rao, Editor Dr. OP Singh, Guest Speaker Dr. Masheri Keshwan Sir, Dr. Thara Ma'am, dignitaries in the audience, and my dear friends, I welcome you all to the webinar organized by Combat Schizophrenia Task Force of the IPS on the eve of the World Schizophrenia Day. The theme of today's webinar is Emerging Trends in Schizophrenia Research, Understanding Causes, Diagnosis, Prevention, and Treatment. And speaker is Dr. Keshwan Sir, who is an authority figure in schizophrenia research. He has more than 700 publications, written 4 books and 100 book chapters. He is editor-in-chief of Schizophrenia
Combat
Research. Schizophrenia I thank you, sir, for taking out time from your busy schedule for this program. Now I would like to invite chief guest of today's webinar, Dr. Gautam Saha, President IPS, for opening remarks and formally inaugurating the CME.
Thank you, Dr. Sandeep Goyal. It's a real proud privilege being a president to organize such an excellent meeting. Credit goes to Combat Schizophrenia team, definitely under the chairmanship of Dr. Sujit Sharkal and full team, Dr. Goyal, you also, Dr. Other, those who are present in this group. Definitely, you have done a wonderful job. And I'm lucky Dr. Thara is there as an advisor, and it's a great, in fact, it is a great motivation to the other committee also, organizing such an excellent meeting. And we are so lucky to have Dr. Mestri Keshavan as a speaker today. We are so proud of him. And on his birthday, it is a really great day for him and for us also. Thank you, sir, again for to be with us. And happy birthday to you again. And I don't want to take much time because everybody is eagerly waiting to listen him. Thank you again, all of you and all the attendees. My regards and sincere love and regards to everybody, all my seniors and my colleagues, my office bearer, Dr. N.N. Raju, Vice President, Dr. T.S.S. Rao, President, Dr. Kirat Mishra, Treasurer, Dr. O.P. Singh, my friend, the editor of Indian Journal of Psychiatry, and also other members of the Dr. Binay Kumar, Dr. Mukesh Jagiala, Dr. P.K. Dalal, all of you, in fact, all seniors. It is really great moment, and I'm not taking much time. Thank you so much. Over to organizer. Thank you.
Thank you, sir. Next, I would like to invite Dr. TSS Rao, sir, to share a few words.
Good evening, and only 2 things. Dr. Macheri Keshavan, when I saw the name, I had no way, any chance of missing it. Our connection from Mysore, in fact, he has a lot of people at Mysore, relatives, and I very, very clearly remember he addressing all the students and staff and postgraduates at our department. And another best occasion is personally wishing him happy birthday. And of course, we are all waiting to listen to. Combat Schizophrenia is one of the very, very important committee in the Indian Psychiatric Society. And a lot of things are expected. Earlier, they have carried out and in the coming days. And I would like to be the participant listening to Dr. Macheri Keshavan. All the best for the program. Congratulations at the outset.
Thank you, sir. Next, I would like to invite editor of Indian Journal of Psychiatry, Dr. O.P. Singh, sir, to share a few words.
Thank you. First of all, happy birthday to sir, Dr. Keshwan. It is a pleasure that you have found time to be with us and enlighten us with this. And I just must say that this is a wonderful program, and with Dr. Thara there, Dr. Sujit as an advisor, EC coordinator, Dr. Sandeep, Dr. Eman, and Dr. Sujit again, and Dr. Kar. So for bringing this program to all of us, and it will enrich us. I will not take much time. We will be eagerly waiting for the lecture. But somewhere, some communication time was 7:30. So I don't know when people are joining, or it will be late. Okay. So all the best wishes, and we are eagerly waiting for Dr. Keshwan to start. So it is a pleasure for us. Thank you, sir.
Thank you, sir. Next, I would like to invite Dr. K.K. Mishra, sir, Treasurer IPS, to share few words.
Good evening, Dr. Sandeep, my great friend, and Surjit Sarkel and Sujit Kar, the task force members behind this schizophrenia task force, and all my office bear IPS, and my best regard to Dr. Kesavan, and wish him all the happy birthday again and again on this auspicious day. And when the Dr. Kesavan, who is in authority in schizophrenia, so I'll not waste time on further discussion. I'll request him to go ahead with his presentation, and I wish the task force all the best on this occasion. Thank you, everybody.
Raju sir is here. Thank you, sir.
Thank you, sir.
I'm sure we will get insight into emerging trends in schizophrenia research after today's webinar. Without further delay, I hand over the proceedings to Dr. Sarkel.
Thank you, Dr. Sandeep. I am given the proud privilege of introducing the chairpersons on this August occasion. Prior to that, I will simply say that we could not have been luckier that Dr. Keshavan, being an authority in schizophrenia, had agreed to speak on his birthday, and that too just a day before the World Schizophrenia Day. And I think I was talking to Dr. Sujit Kar also, and we have personally told all our students and their related, all the people, that you must not miss this lecture, because here we are expecting sir to delve into all the aspects of schizophrenia, right from etiology to treatment, whatever insights we have gained in the recent times in research. And what could be more fortunate than Dr. Thara, who had actually communicated with Keshavan sir to chair the session along with our president, Dr. Gautam Saha. Just a brief introduction. Dr. Thara Rangaswamy is a psychiatrist by training and the co-founder and vice chairman of NGO called SCARF in the city of Chennai, Tamil Nadu. I think we all know. After her PhD on the subject of disability, she lobbied hard for the inclusion of mental disability in the Disabilities Act. She was also responsible for the development of a tool called IDEAS, which we all use to measure disability. She has collaborated with premier institutions around the world, such as Johns Hopkins, UK, and others in Australia, Canada. She has over 120 peer-reviewed publications. She is also on the advisory committee to the Director General of the World Health Organization, Geneva, and was a member of the institute body of the Premier Institute NIMHANS in Bangalore. She is also in the editorial board of several reputed journals, including the Schizophrenia Bulletin. She initiated 2 major programs, an international conference, Icons, and a film festival, Frame of the Mind, and which is going on for the last several years. She has won several important national and international awards, including the President's Gold Medal from the Royal College in UK. She is also an honorary member of the WPA. I welcome Thara Madam as the chairperson. Dr. Gautam Saha, our beloved president, or ever encouraging for everything, including giving us a free hand to organize this program, among other things, is also chairing the session. He is currently the director of Clinic Brain Neuropsychiatric Institute. He is also, apart from being the president of the IPS, he is also the vice president of the SARC Federation of Psychiatrists, and also vice president cum president elect of the Indian Association of Geriatric Mental Health. He is an avid speaker in several locations and also has got multiple publications in national and international peer-reviewed journals. With this, I welcome Gautam Saha sir and Dr. Thara, and I hand over to the chairpersons for further proceedings. And thank you and welcome everybody to this wonderful lecture.
Dr. Thara, please start.
Yeah, thank you, sir. So thank you very much and welcome to all of you, including all the IPS office bearers and Dr. Keshavan, a very good friend of mine for many, many years. It is indeed a very special day today because for the first time, our task force decided to actually start operations with this program. As far as Dr. Keshavan introduction, and if somebody in the audience does not know Dr. Keshavan, that means you are not doing your job well. You need to go and do your reading of all the journals, because anybody who is interested in schizophrenia must know Dr. Keshavan. He's been an iconic Indian American who has created a niche area for himself as far as early intervention is concerned, and also neuroimaging and other biological facets of schizophrenia. I'm going to break convention and I'm not going to read out from his bio summary. All of you know he's a wonderful researcher. He's won many awards. He has over 700 publications, everything. But what I am going to tell you is what I have, my association with him as a human being, as a person, which is not found in either Wikipedia or any other Google search. I have known Keshavan for many, many years. And what has really struck me, apart from all his intellectual capacities, and as I said, has really made a mark in the international arena, is his humility. He's such a humble person, and he is so approachable. Whenever you send him an email, within 24 hours, you'll get a reply, whoever you are. And I think these are hallmarks of a very great person indeed. And I can also tell you that my own younger colleagues in SCARF have been able to interact with him, have been able to get his guidance and advice, although he knew that they were only aspiring young researchers trying to get somewhere. You know, I think this is what every leader should be doing, you know, inspiring people and helping aspiring young people to reach their goals. So Keshavan, it's indeed a great privilege to have you with us here. And indeed, I wish you again a very, very happy birthday, good health in the US, and more accolades in your academic career. Thanks, Keshavan.
Thank you very much, Tara. Can everyone hear me?
Yes, sir.
Thank you very much again, Tara, for those really gracious introduction of me. It's very humbling, and it's indeed an honor to be giving this lecture on combating schizophrenia on the eve of the World Schizophrenia Day during the Mental Health Awareness Month of May, and in the 21st week of the 21st year of the 21st century, if you will. So let me try sharing my slide here. Sorry, let me see here. Can people see the slides? Okay, there you go. Can everyone see the slides? Yep. So, you know, this is an auspicious event of World Schizophrenia Awareness Day. And, you know, I'm going to share my experiences and my current views on how this combat on schizophrenia is being fought. And, you know, I'm one among many of you, one of the foot soldiers of this kind of a combat on this really disabling, devastating illness, which we currently call schizophrenia. So my own journey began, as you see on the top left here, at NIMHANS, and I'll share a few glimpses of NIMHANS with you a little bit later. This is where I am now at Harvard Medical School. Yes. Okay. Did someone say something? Can people see the slides and hear me?
Yes, sir. Yes, sir.
Yes. Okay.
It's
This
okay,
is the
sir.
Harvard
Please
Medical
continue.
School where I work, and this is the Massachusetts Mental Health Center. And, we are also speaking about schizophrenia in this really unusual period of the COVID pandemic, which is affecting all our lives, including the lives of people with schizophrenia, and I'll touch upon that as we go forward. And the key points that I'm going to make are about the causes and mechanisms that underlie schizophrenia, about how we diagnose this illness currently, and how we should be thinking about it in the future.
Keshavan, sir. Shall I send the link? What the speakers for the saying? The WhatsApp. Shall I send it to WhatsApp?
Can everybody mute, please? I request all of you to mute. Yes, sorry, Keshavan.
Oh, that's okay.
Yeah.
So the key points of my talk are on the causes and the mechanisms, the diagnosis and classification or nosology, approaches to prevention and early intervention, management, and the current approaches as well as future directions. Very briefly, schizophrenia, as you all know, is a common illness, about 1% of the general population, commoner than many other neurological diseases, as you see. Schizophrenia is highly disabling, one of the most disabling illnesses in all of medicine. It's a disease that shortens life by approximately about 15 to 20 years. And this is a disease with a high personal and societal cost. It's a disease of theories. As we began learning about this illness, there were many theories, even in my own career. I remember days when schizophrenia was thought to be a disease caused by parents. So one way to think about the theories of this illness is like this fable of 6 blind Indian men groping an elephant in the dark, and each one of them coming up with a different interpretation of what they're experiencing or feeling. If it's a tree or a snake or a spear or a fan or a ball or a rope, depending on what they are perceiving. But turns out that these are all the different aspects of perhaps the same illness, or even that we may not know whether there is one schizophrenia or many schizophrenias. So variously, schizophrenia has been thought to be due to a schizophreniogenic mother, or that it is a spiritual breakthrough and not a nervous breakdown, or that it is a sane response to an insane world, and so forth. But thankfully, a lot of these have been discarded, and we still believe that schizophrenia may be a developmental brain disorder. It may be related to immune mechanisms, and certainly that dopamine may be involved. And I'm going to take you through a little bit of my own journey, beginning in India, so that you'll get an idea as to how I got interested in understanding schizophrenia as a brain disease. I had just finished my neurology rotation, and even at that point, strongly considered neurology as my career. But my heart was in psychiatry, and my professor, K.S. Mani, at that time said, why don't you do some neurological research in psychiatry? So me and my friend Vikram Yaragani, those days there was no IRB. We just took a notebook and we went through the Nimhans Psychiatric Hospital, and we did soft neurological science examination on 100 schizophrenia patients, 100 patients with organic brain disease, and 100 healthy controls. And one of the soft neurological science is what we call the palmomental reflex. You take a key, a noxious stimulus, you scratch the palm, the thenar eminence, and then look at the person's face. It's like a palmist. You look at the hand and then you look at the face and say what's going on. So when you scratch the palm with the noxious stimulus, you see a twitch of the chin. That's called the palmomental reflex, which is a developmental reflex, normally disappears in infancy or early childhood and does not come back later unless there is a frontal lobe disease. So Professor Mani told me, look, you can do these tests in functional psychosis and compare them to organic psychosis, and you will find that functional psychosis will be negative for these tests. And so you can write a paper. This will be a straightforward publication for you. So me and Vikram went around, scratched the palms of 100 people with functional, organic, and healthy individuals. And lo and behold, what we found was that this reflex was positive in 63% of schizophrenics, 65% of organic mental disorders, and 13% of the healthy controls. So we really scratched our head. So what is this so-called functional psychosis? Is schizophrenia an organic psychosis? So that's what started me in the path of understanding schizophrenia as a neurological brain disease, and we'll talk more about that as we go forward. And even clinically at that time, this was very much on our minds. I'll give you an example of one patient. This was a woman who had presented with mood changes, social withdrawal, maintaining postures for long, refusing food. She was immobile, mute, stereotyped posturing, waxy flexibility, classic catatonia. And then we did a neurological exam. So just 1 second, the slide is not moving. There it is. The neuro exam revealed bilateral palmomental reflexes, an increased blink rate, and a positive glabellar tap. These observations and the absence of clear-cut cross-sectional signs of psychosis led to a suspicion of a neurological cause of stupor, and a case conference with neurologists and psychiatrists was held, leading to a detailed medical workup including EEGs and a lumbar puncture. Everything was within normal limits. So anyway, we decided that this is catatonia, and we gave her a course of ECTs. She got dramatically better, and she was diagnosed of schizophrenia, including cognition, as I mentioned, with first episode psychosis showing the most prominent cognitive impairments across multiple domains, with the schizoaffective patients and the psychotic bipolar patients in the intermediate level, and healthy controls there in the green line. And we also see that very prominent soft neurological signs in schizophrenia compared to non-schizophrenic psychosis and healthy individuals. And then, of course, you do see the structural and functional brain abnormalities, which I'll mention to you again in a moment. So what is happening in the brain in people with schizophrenia at a structural level? And one of the important questions for the field is, why is it that this illness begins in adolescence? So it's useful to think through what happens during adolescence. We all have our neurons being born in at the time of birth, neurogenesis. The neurons proliferate, and then the neurons migrate to the appropriate brain regions, following which they prune out, making it a more efficient brain with not too much clutter in there, followed by the remaining neurons being insulated with myelin, the process called myelination, making them efficient conductors of messages, electrical messages. So this process of synaptic pruning happens during adolescence, as well as a prominent aspect of myelination. As you see in this animated figure at the top right, this brain is becoming progressively blue, suggesting a progressive thinning of the cerebral cortex, with about between 20 to 40% of synapses getting pruned out during adolescence. And that was an observation that was made in the 1970s, but it is not very clear what its connection to diseases like schizophrenia is. So Irving Feinberg, a visionary psychiatrist in the 1980s, early 1980s, made a prescient hypothesis that schizophrenia may be caused by a fault in programmed synaptic elimination during adolescence. He thought that the process of synaptic pruning may be aberrant, either too many or too few, or the wrong synapses being eliminated. And he said, we don't know what might be causing it, but it might be a bug in the genetic program. And it might lead to defects of neuronal integration, producing the symptoms of schizophrenia. So this 3 sentences say a lot about what we have learned subsequently about this illness. And he said, if dopamine systems are involved, it may not be central to this illness. So remember all of these, that there may be synaptic problem, and there may be a genetic bug, and dopamine may be kind of a bystander. So this paper, after it was written, was not really much attention was given to it. And a few years down the road, in 1994, we wrote a paper on revisiting his hypothesis. And in fact, Erwin Feinberg told me that nobody else read this paper other than you, and I'm very grateful to you. And so we reviewed the literature in the early 1990s, and we found that all those indices of gray matter reductions and synapse density reductions and metabolic declines and delta sleep loss and prefrontal membrane synthesis, all of those which happen normally during adolescence, are happening at an exaggerated level in schizophrenia. And this is what led us to hypothesize that what happens in schizophrenia is an exaggeration of this process of synaptic pruning, as you see in this red line. And it received support from neuropathological studies led by my former colleague and current chair in Pittsburgh, David Lewis, who showed that there is indeed a synapse loss on these axons in patients with schizophrenia postmortem compared to the healthy individuals. So there is a loss of synapse density, even though we still do not know whether this is due to a failure of synaptic proliferation or is it an exaggeration of the synaptic loss around adolescence. That's a testable question. Then the question becomes, how does the brain know when to prune out these synapses? So that brings us to an important discovery that was made not far from where I work, within walking distance at Harvard, by Beth Stephan. So she discovered that glial cells, microglia, have an important function. Normally, their job is to fight bacteria and fight infection and so on. But when there is no infection going on, they are kind of peacetime soldiers. They're just observing what's going on. And if the brain is functioning fine with robust action potential, then these glia don't have to be doing anything. But if the neurons are not functioning right, they become active and they begin to phagocytose these inefficient or redundant neuronal connections. And they do so by having these complement proteins on their surface, which are in red here, which serve to eat up these synapses, which are giving the message that, I'm not functioning, take me away, and so on. And this is a photomicrograph which shows the phagocytosis and of these synapses. So this is what was discovered in the about 10 years ago. So that was what's happening at the structural level. We'll come back to this point in a few minutes again. So not surprisingly, this kind of a synapse loss might perhaps related to these microglia is happening at the beginning of the illness, as shown by studies of familial high-risk offspring. This is Vaibhav Divadkar, my then postdoc, now professor at Wayne State University. Then the studies of prodromal individuals as shown by Naples studies, showing that there are gray matter reductions during the prodromal phase in people who are at high clinical risk for psychosis. And then, of course, even after the illness begins during the first few years, as was shown by NIMH studies led by Judy Rappaport and Thompson and others. So there is a progressive gray matter loss beginning in somewhere in adolescence or even late childhood. This may point to the structural alterations that exist in this illness. But what about functional alterations? The brain connectivity is also reduced. As you see here, Sean Eek, one of my collaborators, showed that there is a reduction in neuronal connectivity in schizophrenia compared to healthy controls. And these are autistics where there is actually an exaggerated connectivity. See this diminished connectivity. Here is an exaggerated connectivity in autistics, and these are healthy controls. And that connectivity is because of the cables in the brain, white matter tracts that are laid down during adolescence. As you see here, the expansion of connectivity during healthy adolescence, as shown by connectivity expansion that normally happens as the synapses are getting pruned out. The normal thing is for these highways to develop. As you see, when the woods are trimmed out, the highways develop. And that's how it should be normally. But the thinking is that in schizophrenia, the woods are pruned out, but the highways also do not develop. Why might that be at a neurochemical level? At a neurochemical level, the most abundant neurotransmitter system in the brain is the excitatory system of glutamate, which is the most abundant during adolescence. And the other neurotransmitter system, which is also excitatory, which makes its inroads into the prefrontal cortex during adolescence, is the neurotransmitter that is so important for all of us for our risk-taking behavior, and even it's called the love neurotransmitter. It's an excitatory neuromodulator, that's dopamine. So the adolescence is a period with a high activity of excitatory glutamatergic neurons and excitatory dopaminergic neurons, and you might wonder why all teenagers are not psychotic. So evolution has given us the opportunity to balance it out by the inhibitory neurotransmitter, the GABAergic system, also developing during adolescence, so that there is a fine-tuning of the excitatory and inhibitory neurotransmitter systems. So what might happen is that if this fine-tuning of the balance between excitatory and inhibitory systems is haywire, that might lead to a risk for psychopathology. So think of these neurotransmitters as the accelerators and the brakes in our brain. If the brakes don't function right, if the brakes don't develop right, and the accelerator is going haywire, that could lead to an excitatory inhibitory imbalance, and that might underlie psychotic disorders. So let's delve a little bit briefly into what exactly might be the chemical imbalance in schizophrenia. We all grew up learning during our psychiatry residency days that schizophrenia is due to too much dopamine. So that is the dopamine excess theory. And we know the mesolimbic dopaminergic system, and too much mesolimbic dopaminergic activity might lead to psychosis. Straightforward, simple theory. But it didn't explain why people with schizophrenia have cognitive impairments and negative symptoms. So that had to wait for further discoveries. Though we do have evidence now that in schizophrenia, there is perhaps an excessive presynaptic dopaminergic release, as shown by PET scan studies. But it doesn't explain all of this illness. So in order to get a more comprehensive understanding of schizophrenia, one needs to consider other neurotransmitter systems. So the emerging model of this illness is that there is the glutamatergic system, which actually serves to normally regulate the mesolimbic and mesocortical dopaminergic systems. So if there is a failure of glutamatergic regulation of the dopamine systems, that can do two things. One is it can exaggerate or disinhibit the activity of the mesolimbic system, really leading to psychosis. And it can do another thing, and that is it could lead to a failure of another dopaminergic system, namely mesocortical dopaminergic system, which may underlie cognitive dysfunction and negative symptoms. You might ask, why does the person have a glutamatergic impairment in the first place? As you will remember, we just said that the brain is always balancing itself between excitatory and inhibitory neurons. It's like the yin and yang of the brain. So there is the GABAergic system, which regulates glutamate. And if there is a GABA failure, perhaps due to a hypofunction of the glutamatergic receptors on the GABA interneurons, the so-called NMDA receptors, or you might remember it as no more dopamine receptors. A failure of this regulation by GABA of glutamate would lead to inefficient, sometimes exuberant glutamatergic activity, which would then lead to downstream alterations in dopamine, mesolimbic and mesocortical systems. So this is our current thinking about the chemical imbalance in this illness. Then the question is, why does this chemical imbalance happen? We have known for over a century now that schizophrenia runs in families with a high heritability of over 80%, or between 70 to 80%. Until recently, we didn't know what genes might be at fault. But thanks to the discovery of the genome and then the advent of techniques like genome-wide association analysis, here is a Manhattan plot. This is the skyline of Manhattan with the multiple skyscrapers, and you see the Statue of Liberty there. Genetics research uses this kind of a Manhattan plot to depict what genes might be abnormal. So if you take the horizontal axis as the different genes and the vertical axis as the degree of significance to which they are associated with a given disease, and there is this p to the power of 0.008. That is the red line, and all these skyscrapers that go above that threshold line are genes associated with schizophrenia. And beginning in the 1980s, more and more genes began to get discovered for schizophrenia. Now we know about close to 350 gene locations associated with schizophrenia, and it's still growing depending on the sample size. But interestingly, one brain region, one genetic region, kept on growing with each sample size increases. You can call it the Manhattan, the Empire State Building of Manhattan. So people were wondering, what is this particular signal on the chromosome 6? And it was found that this particular Empire State Building may be related to immune genes. And more interestingly, among these immune genes, one particular set of genes, which we referred to already as complement protein genes, complement genes were among the most robust signals here. Also, we know that about at least a third of these genes had something or the other to do with glutamate and brain development. So based on these observations, Steve McCarroll, a geneticist, again within walking distance from my office, he looked at these complement proteins in postmortem brains of schizophrenic patients, and medical students Ashwin Shekhar, along with Steve McCarroll's group, discovered that protein C4A expression is increased in people with schizophrenia. So here is a kind of a detective story. You started off with synapse abnormalities, excessive synaptic pruning, which might lead to excessive gray matter loss, which in turn may be related to complement genes, leading to microglial overexpression of complement proteins and therefore the phagocytosis of synapses that has exceeded the normal threshold. And he also found, Steve McCarroll, that the more number of these complement genes that are transmitted, the higher is the risk for schizophrenia. But then we have to be a little bit humble here. We know that genes are important in schizophrenia, and complement genes perhaps are very interesting and important. But remember that these genes can explain only about 5% of the known variants of this illness. We know that there are hundreds, if not thousands, of genes. There are some genes which have large effect, which are rare, and a large number of genes that have a small effect. Common genes of small effect and rare genes of large effect. And then, of course, there is a large number of unknown environmental factors and gene-environment interactions. So if we are discovering genes of importance in schizophrenia, they probably explain only a small part of what we call schizophrenia. So we can keep that in mind as we go next to our other understandings of this illness. Now, genes and synapses and dopamine, glutamate may not explain everything. This emerging that schizophrenia may be related to increased brain inflammation. This is Paulo Lozano, one of my residents, who my internet connection is saying it is unstable. Can people still hear me and see me?
Yes, sir. Yes, sir.
Yes, sir.
Yes, sir.
That's good. Okay. So Paulo, went into the 1920s literature, and he found that very curious observation that people had made, that the choroid plexus in the brain might be pathologically enhanced or abnormal in people with schizophrenia post-mortem. So we had a sample of about 1,500 schizophrenia patient brains with MRIs, and he showed that schizophrenia patients and schizoaffectives and psychotic bipolars, they all had a elevation of the choroid plexus volumes. Choroid plexus is very interesting because it is the location for neurogenesis, CSF production, immune surveillance, removal of waste products, and supply of nutrients for the brains of people, individuals. So Paulo found that not only is choroid plexus increased, that the higher the inflammatory indices, the more the choroid plexus was increased in size. So this is an emerging view of research, emerging theory of schizophrenia, that there might be an abnormality of the choroid plexus, which is essentially the largest part of the blood-brain barrier. And if there is a leak in the blood-brain barrier, it might allow pathogenic proteins into the brain, leading to brain inflammation. So this is, again, a theory that needs to be further investigated. Now, how might synapse dysfunction cause disability? As you see in this animation here, when one neuron fires and the other neuron gets activated, there is this neuronal dyad that is firing together. And we have known for over half a century that the more the neurons fire together, they wire together. And that process is what is called plasticity or synaptic plasticity. So a question comes up as to whether in schizophrenia there might be not only a failure of synaptic function, but also a failure of synaptic plasticity. Urvaksh Mehta in our group, who was doing a fellowship in my department at that time, he did a meta-analysis of various transcranial magnetic stimulation studies, which is a good measure of brain plasticity. He found that there is actually a blunting of neuronal response to stimulus in schizophrenia, suggesting that there is a failure of plasticity. And that failure of plasticity may be related to this excessive synaptic pruning, which leads to the function of the brain. If you think of brain as a muscle, the brain is not really developing as we use it. So it's like one explanation for schizophrenia is that it is not learning from experience, and therefore there is a disuse atrophy. So this is another emerging view that has to be investigated further. Our patients often ask the question, Doc, you can't make up your mind. You say I have schizophrenia today, and tomorrow you're telling me I have schizoaffective disorder, and next year you tell me I have schizoaffective disorder, and so on. What do you really think I'm having? So symptom-based diagnosis that we make in DSM, like schizophrenia or PTSD or bipolar disorder and so on, have their limitations. They may not really be telling us what is underlying the brain disorder that we associate with schizophrenia. So in order to answer that question, we are part of a large consortium that is investigating the question as to whether there might be multiple subtypes within psychotic illness. So we used machine learning to reclassify psychotic disorders into agnostic categories based on biomarkers. And in doing so, we identified 3 biological subtypes, one of which was characterized by diminished neural response, which we call biotype 1, with prominent cognitive impairment, strong family history, and one subgroup with an exaggerated or noisy neural activity, which we call biotype 2, which may be related to brain inflammation, may be related to neural overactivity, and then a subgroup which did not seem to differ from healthy individuals in terms of these biomarkers, which we have called biotype 3. We'll talk about it a little bit down the road again. So, can we validate this? We have found that using imaging measures that were not used in the construction of biotypes, which were mostly used based on EEG measures and cognition, Cynthia Guimond, a postdoc in my group at that time, looked at the hippocampus, and she found that hippocampal abnormalities were dramatically more prominent in the biotype 1. And the biotype 2 and 3 did not seem to differ. Whereas the hippocampus really did not distinguish between the DSM categories of psychosis. Sinead Kelly, who was another postdoc now back in Ireland, she looked at the biotypes and found that biotype 2 was characteristically associated with elevated brain water, which is an index of brain inflammation. So I've told you so far about brain biology, but this is a talk about all aspects of schizophrenia. So I'm going to take you through a little bit of what's happening in terms of psychopharmacology, as well as psychosocial treatments. So I don't need to teach this group more about antipsychotics. We all grew up with learning about the first generation antipsychotics, which are mostly, characterized by their risk of producing extrapyramidal side effects. Then came the so-called second generation antipsychotics, such as risperidone, olanzapine, and quetiapine, and so on, which are characteristically free from extrapyramidal side effects, even though some of them do cause EPS in higher doses, like risperidone. Some of them also produce metabolic side effects like sedation and weight gain, and risk of diabetes. And then we have the so-called third generation antipsychotics, which have an effect not only on dopamine and serotonin blockade, but also serve like partial agonists, some of these neurotransmitter systems. Aripiprazole, brexpiprazole or Rexulti, which may have some antidepressant effects, cariprazine, which is which may be good for negative symptoms. These are all still to be determined. I don't know how many of these are available in India. Lumateperone, Caplyta, one of the most recent of these, which again has a serotonin, dopamine, and glutamate effects, may be somewhat good for negative symptoms, and so on. This slide is a summary of the side effects of the various compounds. You see a lot of red in the first generation, which have both EPS, extrapyramidal side effects, as well as metabolic side effects and anticholinergic effects and weight gain and so on. And then you also have some of these older atypical or second generation drugs like clozapine and risperidone and olanzapine, which have a lot of red here with more of the metabolic side effects. But some of the more middle ones here, which are relatively side effect neutral, are the more recent ones: lurasidone, brexpiprazole, asenapine, and aripiprazole, and so on. So every year there is one or other newer Me Too compound which works on dopamine that is being approved by the FDA. But there are other compounds that are outside of dopamine, which I'll come to a little bit down the road. Let me take you through a little bit about another important paradigm shift that's happening in the field of psychotic disorders, namely early psychosis intervention. There is the emerging view that schizophrenia can be identified early, even in its prodromal phase, the so-called individuals at clinical high risk, and then they can be followed up, and a good number of them can be predicted if they are likely to develop psychosis, about a third of them. And about a sixth of them don't develop anything, but sadly, about half of them develop some problem or the other, some other psychopathology or maybe a functional impairment. So this is an important question to study. How can we predict outcomes in people who are at risk for psychosis, and then prevent psychosis and prevent other negative consequences? Earlier, it was thought that atypical antipsychotic drugs in low doses like risperidone or olanzapine might be a good idea, but then their side effects can limit any potential benefits. More recently, there is some promise that some psychotherapeutic approaches like cognitive behavior therapy and family-focused treatment may have modest benefits. Perhaps cognitive remediation may have a modest benefit as well. Studies are beginning to show that psychosis conversion, risk for psychosis, can be predicted using clinical and cognitive predictors using a calculator. This is Ty Cannon in the Naples study, who showed that a risk calculator predicts conversion to psychosis in about 70% of the time. So this is the curve, Kaplan-Meier curve, the ROC curve showing the predictive value of the Naples calculator. In our own group, Bill Stone has replicated this in another sample that we collaboratively collected from China. And Jaya Padmanabhan in our study, in our group, looked at the calculator, a different calculator, and found a predictive value of over 70% for familial high-risk individuals as well. So psychosis can be predicted. There is an emerging view that clinics that are specifically designed for first episode psychosis patients, the buzzword is coordinated specialty care programs that involve a team-based approach to include not only psychopharmacology and individual psychotherapy, but also peer support, cognitive remediation, family-based treatment, integrated medical and behavioral and substance abuse care, and supported employment. A combination of these, as opposed to any one or more of these, could be more effective than routine treatment. And indeed, that was shown by this large study by John Kane and others called the RAISE study, Recovery After Initial Schizophrenia Episode. And what was found was that symptoms and quality of life were both better in the coordinated team-based approach to care compared to treatment as usual. And it was also found that those with shorter illness duration tended to have better outcomes than those who had already had a treatment delay of over 74 weeks. And a recent meta-analysis by Christoph Korrel showed that across several randomized trials, multiple indices of outcome, including remission, recovery, positive and negative symptoms, and global functioning and quality of life, were all better with early intervention models compared to treatment as usual. So this is an important shift in our approach to treatment of schizophrenia. What about treatment resistance? The only drug that we know which is superior to standard antipsychotics that works for treatment-resistant schizophrenia is clozapine. You all know that. But the one important question for the field is, how does clozapine work? What is it that makes clozapine superior compared to the other antipsychotic drugs? So that has remained poorly understood. One theory is that it may have something to do with D3 receptors. Another theory that it may be related to muscarinic receptors, M1, M4. There is a third theory, which is we are very interested in it. We did a meta-analysis, we did a systematic review and found that one interesting aspect of clozapine is that it increases slow wave activity in the brain, especially in the theta range and somewhat in the delta range as well. So you all know that clozapine in higher doses causes seizures. But in therapeutic doses, it doesn't cause seizures, but simply increases slow wave activity. So we are interested in the question as to whether clozapine's effects on EEG used as therapeutic bioprediction outcomes. That funded to do, and over the years we hopefully will be able to answer that question. Moving on to psychosocial treatments, we already know, and as I said before, that cognitive impairments are at the center of this illness, underlie the disability of this illness. But the unfortunate point is, even though we know multiple neurotransmitters are involved in cognition, none of the medications that work for any of these neurotransmitter systems has any established therapeutic benefit for cognitive impairment in this illness. What does work is brain training. We talked about brain plasticity. The more you use the muscles in the brain, the better they get. And that is what takes us to the whole idea of cognitive enhancement or cognitive remediation. And we have been developing an approach to cognitive enhancement therapy, cognitive remediation called CET, or cognitive enhancement therapy, which is a combination of neurocognitive remediation using computer exercises along with group-based social cognitive training. So that involves individual coaching, computer-based exercises. Nowadays, we are doing it remotely in the COVID era through Zoom and group sessions with homework. What we have found in 3 independent controlled trials that chronic schizophrenia patients early course schizophrenia patients and another second independent cohort of early psychosis patients, CET is better than routine supportive treatment. Not only that, we have found that with CET, there is a reversal or protection against gray matter loss in key brain regions like the amygdala and the hippocampus and the cingulate. So, based on the randomized controlled trial results, we have now launched a implementation study across 19 sites in New England to see whether CET can be implemented in community settings. We have also been looking at whether physical exercise can enhance cognition, and we are developing a virtual reality-based approach to enhancing cognition while the person is doing exercise on a treadmill, solving cognitive puzzles in virtual reality. And so this is a direction for the future. Another direction is to use sleep as our therapeutic window. You all know that when you learn something and sleep over it, you remember it better the following day. This memory consolidation happens during sleep, and that happens because of a particular oscillation in the brain called spindle oscillations. These are called spindles because they look like spindles. And spindles can be enhanced by transcranial magnetic stimulation during nap. So that's a study that we are hopefully going to be doing in the near future to see whether triggering or enhancing sleep spindles can enhance cognition. So this is Bob Stiggold, a world-renowned sleep researcher who is in our group as a leader in these studies. Another approach is to use remote cognitive remediation using smart devices. John Torres in our group has been leading this. He is developing an approach to what he calls digital phenotyping by collection of multiple domains of information on symptoms and cognition, physical activity, and sleep parameters on a 24/7, 365 basis using smartphone active and passive data. And it's possible to also introduce cognitive interventions as well as other interventions like CBT using smartphones as well. Roscoe Brady in our group has been developing ways of enhancing cognition as well as improving negative symptoms using a tract-based modulation of specific brain tracts that are underlying different dimensions of symptoms. For example, he showed that cerebellofrontal tracts are related to negative symptoms, and by specifically targeting the cerebellum, he showed that negative symptoms can be improved in schizophrenia. Now, this is Mullah Nasruddin, a Sufi character, mythological character, who, not mythological, he was a real person, who pointed out to the futility of looking for a key where the light is, whereas the real key might lay somewhere else in the dark. So the point here is that just because dopamine blockers work for psychosis, it doesn't mean that the etiology of schizophrenia lies in dopamine tracts. So we have to be looking for alternative targets for treatment and alternative explanations for etiology outside of dopamine. So that brings us to the various targets that one can utilize in developing novel therapeutic interventions outside of dopamine. For example, if this glutamatergic synapse, one could use glutamate modulators, nicotininergic agents, serotonergic agents, cannabinoid agonists, GABA modulators, anti-inflammatory and antioxidants, neuromodulation, and as we have discussed, cognitive remediation and so on. So glutamate itself is an important target neurotransmitter. And a number of studies are now looking at whether glycine, which is an agonist at the NMDA receptors, could be beneficial therapeutically. Glycine transporter inhibitor, which has the same effect of increasing glycine. Bitopertin was tried without much success. Sodium benzoate which enhances glycine through enzyme activity. It's an enzyme blocker of the breakdown of glycine, is being tried. mGlu agonists, metabolic glutamatergic receptor agonists like pomeglometad, a Lilly compound, has been tried with some mixed benefits. Lamotrigine, Lamictal, which releases glutamate, is being tried. Phosphodiesterase antagonists, and so on. So the whole field is quite active, and we still do not have good, clear answers. Among other neurotransmitters, serotonin, an interesting compound, pimavanserin, Nuplazid, don't know if it is available in India, was approved for psychosis in Parkinson's disease. It's also pending for dementia psychosis. Some studies are beginning to emerge showing that pimavanserin might help negative symptoms in schizophrenia. Cholinergic compounds have become particularly interesting. There is a muscarinic M1 agonist by name xanomeline, and when combined with a peripheral anticholinergic, this is a drug that is investigating xanomeline along with trospium in psychosis using by the Karuna Pharmaceuticals. And they found, this is a paper that came out last year in New England Journal of Medicine, showed a fairly robust reduction of psychosis with xanomeline trospium compared to placebo. So this is probably one of the first non-dopamine antipsychotics that is beginning to show some promise. Cannabidiol is another potentially interesting compound, and also works on GABA and glutamate balance. Phil McGuire showed that it has a beneficial effect in schizophrenia, but a Yale study showed negative results. So the jury is out. We don't know yet. Another interesting direction is the so-called TAR receptors, traceamine associated receptors. And again, another New England Journal paper showed that last year showed that PANSS scores are reduced significantly with the TAR1 agonists compared to placebo in schizophrenia patients. And I have mentioned inflammation as a potential target. Anti-inflammatory agents like metformin, minocycline might have a therapeutic benefit, and antioxidants like N-acetylcysteine may have a therapeutic benefit as well. Another recent development is the introduction of valbenazine as a treatment for tardive dyskinesia. It works on vesicle monoamine transporters, so VMA inhibitor. It's a drug that kind of is an offshoot of tetrabenazine, which was an offshoot of reserpine, which of course Indians knew about its value in psychosis going back thousands of years. But reserpine causes suicide risk and depression. So did tetrabenazine, but valbenazine is supposedly free from that. Now, very briefly, this is the COVID era, and I want to mention briefly about the impact of COVID on schizophrenia. It was reported in a paper in January in JAMA Psychiatry that schizophrenia in a large study in New York is second only to age as the highest risk factor for dying out of COVID-19. So this is something that we all have to keep in mind as we take care of people with schizophrenia in this day and age. It's possible that this higher mortality in schizophrenia due to COVID may be due to higher medical comorbidity, which patients with schizophrenia have, may be related to less healthcare seeking by these individuals, and also maybe higher disparities in medical care. So more research is needed, and also we need to be vigilant about this. I'm going to end with one other study that we are doing. A number of our patients have frequently, expressed that they don't like to be given the diagnosis of schizophrenia. And we have a consumer advisory board where I work. We have patients who work together with doctors to do research on the consumer perspective. Around the world, there has been a trend towards changing the name of schizophrenia. Like in Japan, it's now called integrative disorder. In Korea, it's called attunement disorder, and so forth. So we asked the question in a large survey of over 1,100 people, including patients, clinicians, scientists, and policyholders, and so on. We found that 74% favor a name change for schizophrenia. And one name that actually was suggested by one of our patients, altered perception syndrome, got the highest marks as to an alternative name. Interestingly, altered thought and perception syndrome is a name that Hong Kong has adopted. Another favorite was psychosis spectrum syndrome. But a lot of other names like Bloiler syndrome or Kraepelin syndrome or aberrant salience syndrome and neuroemotional integration disorder and so on, they got lower marks in essence. Even integration disorder and so on. So among the various names and words that they used in these surveys, in our verbatim responses, stigma was the largest number in this word cloud, as you see. So which really is a big thing that we have to combat in our quest for combating schizophrenia. To me, the biggest challenge is stigma, and we have to be fighting that in this 21st century. So let's see here. Yeah, that's my last slide. Schizophrenia might be an illness that results from an exaggerated synaptic pruning during adolescence, which in turn might lead to a deficit in brain plasticity. Dopamine is proximately involved, but imbalances in glutamate, GABA, and other molecules and psychotic disorders result from polygenic interactions which affect brain development, synaptic integrity, and perhaps immune functions and channel functions. Developmental brain abnormalities cut across symptom-based diagnosis, and there may be distinct biological subtypes with distinct treatment targets. Emerging observations set the stage for prediction and novel interventions in populations stratified by neurobiology. Future treatments include glutamate, GABA, and cholinergic agents, anti-inflammatory and antioxidants, targeted neuromodulation, and psychotherapy. Schizophrenia remains highly stigmatized, and many stakeholders prefer a name change. So I think that's my last slide, and I want to thank all of you for your attention. And also, these are my various sources of funding and disclosures, and we're grateful to our patients and their families for supporting us, and There are many of my collaborators, too many to name, that have helped us in these studies. Thank you all.
Thank you, sir.
Thank you,
Thank
sir.
you, sir.
Thank you, sir, for the
Thank
wonderfully
you.
comprehensive presentation.
Yep.
So there have been
Thank
a lot of questions, sir. So too many questions on the classification, diagnosis, outcomes. So one of the first questions, sir, how widely implemented are the early intervention in psychosis programs in the US?
Yeah, that's a very good question. And, thanks to a governmental action which led to more funding, there has been probably over a tenfold increase in the number of early psychosis clinics around the country now, with the latest around 250 early course psychosis coordinated specialty care programs in the US. So there is a fairly rapid expansion.
Okay, sir. Sir, another question is with reference to the sleep study that you mentioned. What was the methodology adopted for inducing sleep?
Well, we have not done that yet. I just mentioned it as a future direction, but there are other people who have done these studies. And so the approach is one could do it in two ways. You could use transcranial magnetic stimulation, or TMS, or you could use transcranial direct current stimulation, or TDCS, applied to dorsolateral prefrontal regions. Which with these techniques you can't go really deeper than that. Ideally, you would want to stimulate the thalamus, but studies have shown that during non-rapid eye movement sleep, and after a person falls asleep, let's say during a nap, you can use neuronavigation to identify the key brain regions and use these two approaches, one of these two approaches, to trigger both delta sleep as well as spindle sleep.
So speaking of RTMS, another question was on the protocol used for RTMS in improving negative symptoms.
Yeah, so, even though others in my group have done this, like Roscoe Brady and Urvaksh Mehta in India, I am not, immediately familiar with the specific details of this. I think Roscoe Brady uses 2 sessions of 20 minutes each daily for 5 days for getting the results that he gets of RTMS.
So do you see interventions based on genomic manipulation? Can they see the light of the day for schizophrenia anytime in the future?
I'm not very optimistic about that, because the way we think about schizophrenia, it's probably not related to one single gene or even a small number of genes. And it's more likely a result of polygenic effect of hundreds of genes. And genomic manipulation at this point may be maybe not a very fruitful approach, with the exception of this approach called precision medicine. That is, if you identify families with a specific mutation, and then you introduce an intervention that specifically addresses that particular mutation, there may be a benefit. For example, Deborah Levy at the McLean Hospital in Harvard has been studying one particular gene that has an impact on glycogen synthase, and using a dietary manipulation, she has shown results of relief of schizophrenia symptoms in that person. So, but those are rare. These kinds of mutations, there are other similar studies that have been reported using other other specific genetic alterations and so on. But if you consider schizophrenia as a mountain to climb, these are all one stone here and one stone there. Scaling the schizophrenia mountain, is with single gene manipulations is not likely to solve the problem.
There are a lot of questions on the course and outcome in schizophrenia. So it's like how over the years, how much has the treatment outcome changed? How much recovery is possible in schizophrenia, both statistic as well as functional? And would you consider that these days the prognosis is better? So your thoughts on that, sir?
I think so for two reasons. One is, my own personal experience over the last 35 years of taking care of these illnesses. I've seen that the outlook has changed. We don't associate schizophrenia as this kind of a, being sanctioned to a lifelong illness and so on. We have increasingly seen a tremendous amount of heterogeneity in this illness. And over the course of the last decades, I can say several examples of people who I thought will never do well early on in taking care of them. And then I've been surprised to see people who are professors in universities who come and introduce themselves and say, you took care of me. You said I had schizophrenia. Of course, they still have schizophrenia. They're taking medicines, but they're doing okay. So what I tell my patients these days is, unlike many neurodegenerative disorders, in schizophrenia, probably the worst aspect of the illness is in the early few years. And the majority, there is some kind of recovery that happens over the course of time. Some more, some less. But, therapeutic alliance is key and illness management is key. So, that's my experiential account. From a research standpoint, there are no clear-cut data as to whether the trajectories have changed over the last 50 years or so and so on. But we know two things. One is the longer the illness goes without treatment, the longer the so-called DUP or duration of untreated psychosis, the poorer the outcome. This expansion of early detection strategies and early intervention is likely to favorably impact the overall trajectory if it is implemented right in large scale across multiple populations. So we don't know whether indeed it has changed the trajectory, but that's the hope. Yeah.
So the role of psychedelics in the treatment of schizophrenia, particularly for negative symptoms.
Well, I get that question all the time, but I don't have a good answer for that. This is really an emerging field as to whether psychedelics like psilocybin or MDMA or even ketamine, whether they have therapeutic effects. The only thing I've seen in some level of evidence base is the use of psilocybin in a controlled trial, which was published in New England Journal of Medicine a few months ago for depression. But and of course, ketamine, which is which used to be a drug of abuse at some point, it does work for depression. But in schizophrenia, there is no data, to my knowledge, of any of these compounds like psilocybin having been used successfully.
Okay, sir, the final rather comments, not question, by Isaac, sir. He mentions that a rose by any other name would smell as sweet. Your comments, sir.
Sorry, say it again. A rose by any other name
would smell as sweet. So your comments on that, sir, about the name change, I presume he was talking about that.
Yeah, so that's a very, very thoughtful point. Is it Mohan Isaac? I guess, I think it is Mohan.
Yes.
Yeah. I agree with you, Mohan, and this has been a criticism of any name change, that even if you change the name, it'll regain whatever its positive or negative value down the road. But my response to that is that we don't know. It may well be that any new name will also become stigmatized. That's quite possible. However, at least let's make a choice to give the disease a name by what it actually is. We don't have a very good answer to that, but we don't know. We don't believe that it is a split brain disease or split mind disease. It's clearly a misnomer. Is it a disease that is primarily a dopamine dysregulation disorder? Probably not. Is it probably a disease that is primarily an integration disorder in the brain? Yes, but people don't fully understand what that means. Is it a psychosis spectrum disorder? It's probably more true to say that because it is a spectrum. It's not one disease. Psychosis can also get stigmatized. The word psychosis can get stigmatized. So there is no good answer to this, but certainly we have an unsatisfactory name at this point.
Thank you, sir, for the patient elaboration. I would like to hand over to the chairpersons for further proceedings.
Thank you, Dr. Keshavan. It was really nice. It is another one presentation. We are hearing a great presentation by you. It is elaborative and extensive and on original works by you also, and it is really fantastic. I think we learn a lot today from your presentation. I think people will agree with you. And one thing I want to know, maybe Dr. Keshavan is a very good, as an academician, Dr. Thara told that if you are not aware of it, it is your fault. Nothing can be done, but he is also a very good artist. His painting, I am a very big fan of his paintings. Usually I saw in the Facebook. Very good artist, and it's really, being a modest human being, he is a very good artist. Thank you, sir.
Thank you.
Dr. Thara, please.
I'm fine, Dr. Saha. Once again, thanks a lot to Keshavan. I think it was an exemplary talk, so multifaceted that you're able to cover just about everything, including antipsychotics. So thank you once again, and thanks for agreeing to do this on your birthday. Spending 1.5 hours on a very special day. Thank you so much.
This is the best gift I got for my birthday.
Oh, thank you.
Thank you, sir. Thank you, sir.
That's very nice to hear.
A return gift for us, sir.
Thank you.
So, good evening, everybody. Now, this is the time for vote of thanks. This is, I will say, this is a wonderful day for all of us who participated in this webinar. First of all, I want to pay my sincere regards and thanks to Macheri Kesavan sir, who gave his precious 1.5 hour time and on this precious day, on his birthday. And we are very much thankful to you, sir. Always it is a pleasure to hear you in the audience. And today we have 234 participants, which itself says that what is the impact of your presentation, sir. Thank you so much for giving us this time. I'm sincerely thankful to the Indian Psychiatric Society for giving us this opportunity to conduct this webinar on this eve of World Schizophrenia Day. Thank you so much to Gautam Saha sir, TSS Rao sir, KK Misra sir, and OP Singh sir for giving your precious time for this webinar. This program will remain incomplete without my paying the regards to the delegates who participated in this webinar. 221 live users at this moment. It says that a lot that what is the success of this program. So thank you all the delegates for attending this program. My sincere regards and thanks to IPS Combat Schizophrenia Task Force members who guided to reach this point. Thank you, Thara ma'am, for guiding us. And last not the least, my sincere regards to Sun Pharmaceuticals for creating this digital platform and doing all the technological support for conducting this webinar. Thank you. Thank you, everybody.
Thank you, sir.
Yes.
The recording has stopped.
Thank you, Sujit.
Thank you so much, sir.
Wonderful.